If you're just tuning in there, this is the Okeanos Explorer. We're diving today at Keliʻihananui seamount. That's a proposed name here in the Central Pacific Basin, the Line Islands region. And we're just getting the bottom into view here. We're currently at a depth of about 1,920 meters, and we're about 20 meters or so off the bottom. If you're watching on camera 1, you're looking at our Deep Discover vehicle, D2. And if you're watching on camera 2, you can see the view from Sirius, which is another ROV that's attached up above D2. That's what D2 is tethered to. And then if you look over on camera 3 today, you'll see sort of a quad view that'll have our navigational maps and lots of other information about what's going on. All right, so let's take a little bit for the pilots to get settled in here, and we'll get our first good views of the bottom.
From this bird's eye view, it looks like there's a fair amount of sediment. I think I'm seeing a shrimp swim across the bottom slowly there.
Sorry, can I change this?
There's even more sediment than I expected from that bird's eye view.
Right.
Looks like it's all sediment. You can see, I think the darker parts are probably aggregates of organic material that have settled from the surface and get caught up in these little depressions. And I'm seeing, for example, there's a brittle star coming in the upper right side of the screen, and it's next to a structure. And we'll have to see when we get to the bottom, but there's these single-celled organisms, xenophyophores and Camochiaceans, that build these structures out of sediment. They have their cell cytoplasm on the inside, and other organisms tend to aggregate around those structures, especially the small infaunal material, the small crustaceans and worms that are much too small for the cameras we're seeing. They're microscopic, but they would live in the sediment. Go ahead, pilots, watch lead. Outstanding, thank you. So are you guys in a position that you're ready to go? Why don't you give yourselves an introduction first, and then we'll get on our way.
All right. Well, in pilot today, I am Andy Lister, and I will let my co-pilot and navigator introduce themselves, but we are all with the Global Foundation for Ocean Exploration, or GFOE.
Thank you, and Caitlin Bailey was here in the back row. She just stepped out for a moment. Why don't we start by having a look at this elongated thing right in the center of the screen. Thank you. See, Les Watling from the University of Hawaii has noted online that there's a lot of organic fluff on the bottom, and he says that's a good sign, and I think what he means by that is there should be plenty of productivity to support biology down here. Looks like that's probably an old fecal trace. So we have deposit feeders like sea cucumbers that sort of move around and munch on these sediments and extract all these dark-colored organics from them, and they leave a fecal trail behind them, and it's one way. There's a whole set of studies. Maybe you're familiar in geology. You know, I think they look at some of these fecal traces in the fossil record.
That's right. Yeah, we look at bioturbation and in tubes, and there's a whole field of research there in the rocks.
Right. The bioturbation is important for overturning the sediments, and is important to understand if you're going to try to age any of that rock, I guess. Mm-hmm. Okay, pilot. Thank you. We can begin our transit.
Yeah, in some cases, some of these soft-body animals, that's the only fossil evidence, these sorts of traces in the rocks. So.
And, you know, a couple of dives ago, maybe it was 2 dives ago, I think, we got kind of excited because we saw on the hard bottom, there was a place that was much brighter, and you could see that something was making a trail. And we've seen that very often. It's an indication that something is indeed kind of munching its way over the bottom, and we were lucky enough to see a chitin.
Yeah, that was pretty cool.
Right, so often we just see the trail, or we just see the fecal mound or the fecal spiral, but it gives us some clues to what may have been here before. Astrid Leitner is asking, what is all the fluff? And I suspect most of this fluff is just marine snow, organic particulates that have settled on the bottom, so the remains of the phytoplankton that have been living up in the shallower parts of the water column where there's light, or fecal material that's raining down, dead organisms, shed exoskeletons, all that sort of organic material. Eventually, if it's not eaten in the water column by the various organisms that we would see on today's midwater dives, midwater transects, it's going to settle on the bottom, and it kind of accumulates in the little depressions in the sediments.
Yeah, we're not seeing ripples today, at least so far, like we have some of the other dives. So maybe we'll hear from the pilot a little bit later if he gets a sense of the currents, but at least initially it looks like it might not be as strong as some of the other places.
Can we get a zoom just next to the lasers? I see something colored there. Another great example of a coiled fecal mound. So that would be from a holothurian. They have these twisted intestines.
Wow.
And I think this is a little cup coral there. Oh, nope.
Oh.
Cup corals don't move like that.
That's a crab with an anemone on it, huh?
Exactly. So that's a hermit crab, a pagurid crab. And I think behind it is probably one of these xenophyophores that we're talking about, these single-celled organisms that can build these plate-like or ball-like structures. So there we go. This is a...
Oh, he's on the move.
He's on the move. Hermit crab that probably had a snail shell that it was tucked into, and then it got an anemone on that shell, and the anemone has now grown to completely block the shell itself. And it may be, there are some species of these anemones that continue to secrete a shell so that the crab doesn't have to get a new shell, it can just grow along with the anemone. Thank you, pilot. Les Watling is suggesting that the evidence of the bloom that we're seeing may be an indication that the evidence of this dark material that we're seeing on the bottom may be a suggestion that there was a recent bloom in the shallow water. That is, we've had a kind of a population explosion. It's a good time of the year. There's lots of sunlight. We have some renewed nutrients at the sea surface. Lots of phytoplankton get produced, and once they have died or they've been eaten, the remains settle to the bottom. And you would expect that this would be consumed fairly quickly. There's a lot of organisms really searching for food down here in the food-poor deep sea, so it shouldn't last all that long. So if we see so much of it, it might be an indication that it was a relatively recent settling. Understood, pilot. Thank you. Great. So we're beginning our move to head up slope.
All right. We'll be coming up on a little bit steeper topography a little bit later. You know, one of the things I've enjoyed in these dives, just seeing how things change often with just a break and slope we see.
Yeah, the white
different animals.
This looks like a polychaete worm. It's kind of swimming through the water column. So these are benthic animals that every once in a while will move up into the water column, and it's related to an earthworm. It's an annelid, but you can see it has these extensions from the side of the body. Each segment has one of these extensions. They're called Parapodia, And you can think of them as kind of paddles, if you want. And they look kind of shiny and iridescent. They're catching the light. They're actually a series of bristles. So it's kind of like a mini broom sticking off of each side of that worm.
So looks like he's doing the backstroke there.
He's doing the backstroke, and now he's going to settle back down and feed on the bottom. Thank you, pilot. Tim Shank agrees with Les's observation that he's seen at about 2,000 meters deep on the seafloor near the Galapagos in the spring similar accumulations of these organic materials. This is a really good indication. We've talked about this, and it's one of the interesting concepts about capstone that we're investigating, and that's the linkage between what's happening in the shallow water and what's happening in the deep sea. So much of this brownish material, or slightly off green, olive green material you're seeing, originated at the surface of the ocean and then has settled down here and is providing food for the animals that live in the deep sea. So there's a really tight connection, even though we say that there's no photosynthesis in the deep sea, and so there's no production of food in the deep sea. It's still reliant on what's happening in the shallow water. Looks like a shrimp swimming through, and I noted when we were sitting on the bottom, we saw something swimming, and I'll bet it was a shrimp, much like this one. Just briefly, we see this guy quite a lot. So you can see it's very large swimming legs. Those are called Parapodia, pleopods. Parapodia was in the worm. Those are pleopods. The common name is swimmerets. And one of the things you can see on this particular shrimp is that those swimmerets are being sort of kicked out to the side of the animal instead of beating directly underneath the animal. And that's one of the clues to its identity. I believe that's in the family Aristiidae. You can see very, very long antennae trailing behind it. It's basically using that to sense its environment. Keep in mind that this is normally a very dark environment. The ROV has these great lights that allow us to see. Correct. Yep. Thank you. That was just a brief view.
So I think that might have been a Plesiopenaeus, which is an Aristeid.
Great. Thank you, Astrid. Plesiopenaeus armatus is the suggestion. You'll hear us give a lot of these Latin names. It's the formal naming convention in biology for species. And as much as possible, we can try to translate what the names mean. I certainly will not know all of them. But plesio usually refers to something that is old or ancestral. And so I'm going to make a prediction that somebody thinks that this particular genus of shrimp represents a fairly old lineage in that family.
And what characteristics would they use to decide that, Scott?
Well, what's done mostly now is using DNA analyses, and so we would look at the sequence of the genes, select a certain number of genes, or now we have the technology that we can actually get a broad scan of the genome. And there's a variety of computational analytic methods that you can use to build what are called phylogenetic trees. And they're basically charts that link together the most closely related taxa based in their genes. And so then we can start to see patterns in that tree. And if you think of a physical tree, you could say that all the leaves are connected to branches that go back to one limb. So all of those leaves would be more recently connected to the main tree than leaves that are on another limb of the tree. And so that's the sort of thing we're looking for. And in terms of which one is the closer to the ancestor, it's basically when did it arise on that tree. The beauty of using the DNA over the morphology, and I mean, we do use the morphology as well, but the DNA, you can compare a much broader suite of species because not all animals and plants and so on have the same sorts of morphologies. You can't directly compare them.
And sometimes we're surprised, I bet.
We're surprised a lot because what we think we're seeing in the morphology, we may be fooled by. And there's something called convergent evolution. And that is where you have organisms that are living in similar habitats that have independently evolved to similar characteristics in order to deal with the challenges that they meet in those habitats. maybe there's something just went off the left side of the screen. It'll be on the lower left now. Copy that, pilot.
All right.
Copy. I think I saw a holothurian there, a sea cucumber, which is totally what you would expect on a sedimented bottom like this. They're deposit feeders, so they would be feeding on the soft sediment. We've already seen a couple of fecal traces that suggest they're here. Tim Shank has just offered an interesting observation. Oh, look, we have some rocks that have been
something poking
exposed
out there.
Yeah. Here's some more. This is, I think, what we'd expect as we get to the steeper part of this slope.
Right. We may see different animals. We certainly have seen a lot more life on some of these higher rocks on our previous dives. So.
Sure. Tim Shank is just noting that the name Plesiopenaeus is no longer accepted. That now we use the name, let me pronounce this here, Cerataspis. In fact, the whole species name, Plesiopenaeus armatus, is now Ceratapsis monstrosus, and that's based on a molecular phylogenetic study. So exactly what we're saying, we use the DNA and it can reveal different kinds of relationships, and sometimes we move the species from one classification or one name to another.
Oh, great. Thanks, Jim.
Looks like a midwater fish there swimming by, but it looks like this is a crinoid perched on the rock. Actually, maybe that's not a midwater fish. This looks a little bit different. This looks like a little brocculid or something. So Astrid, Basocetus, she's saying.
Hi, yeah, so I actually think this might be a Basocetus species. You can see the really bulbous head. It's probably an ophidiid. This guy looks really small, though, so he might be a juvenile, which makes him a little harder to ID. And he's moving pretty quickly. But yeah, my guess would be Basocetus. That would be the genus. These guys are actually, we've been trying to trap some of these guys for specimens to try to figure out exactly what they are. We see them a lot at abyssal depths, and we've seen them a bit up to 2,000 meters as well in these Okeanos dives. Yep, now that I got a better view, that's definitely a Basocetus. We've been trying to call it Basocetus nasus, but we don't have any specimens, so one day we'll catch one of those guys, and then we will know. But for now, Basocetus.
Great. Thanks, Astrid. So you said ophidiid. That's a cusk-eel?
Yep, they're in the cusk-eels, Ophidiidae.
Great, thank you. And pilot, if you're not too far off course, if we can get a look at this brownish thing in the lower center. Oh, and I think I see an umbellula. Is that what I see? Yeah, no, a sea spider. So directly under the lasers, there's a sea spider. We haven't seen one of those during this expedition yet. They're always fun to look at. And then there's a sea cucumber to its left, but let's look at the sea spider first. This is a big one. It's got some long legs. Oh, and there's an urchin underneath it as well. So you're looking at a sea spider. This is in the group Pycnogonida. And you can see it used to be actually the group used to be called Pantopoda, which translates to all leg. And you can see why. It seems like there's virtually no body. And in fact, some of its organs project into the upper parts of the legs, so parts of its stomach and its reproductive system. Pycnogoda means knobby knees, so that's not a bad name either.
Yeah, descriptive.
And you can see, so the right side is the head end, and there's a long tube that's extending down from the head end, and that's its proboscis. And it can extend that proboscis, or not extend it, but it can bend down, put that proboscis into various soft-bodied polyp-like organisms, so anemones, zoanthids, corals, and it literally sucks out the fluids from inside there. So a really nice observation. And to its lower right, we see one of these Plesiodiadema urchins. And I can't resist sharing with you a quote that I saw on Instagram. We had posted a photo of this urchin. And someone who goes by the tag, "We Are the Ocean Forever," said, "The most terrifying cotton ball of all time." So that's a pretty good descriptor, I think.
Yeah, I'd agree with that.
Yeah, thank you for sharing that. To the left, yes, thank you. I really don't know what that pink wavy stuff is in the background there. Yeah, so here's a sea cucumber. So we were describing deposit feeders. This is an echinoderm. Believe it or not, this is very closely related to that terrifying cotton ball over there, the urchin. They're both echinoderms, which means spiny skin. It's just that this one, the hard parts, that are in the skin are much smaller and distributed throughout the softer part of the body. And in fact, if you think of that urchin, it had its mouth underneath and its anus on the top side of that ball, and if you stretch it out and lay it on its side, now you have a sea
There you
urchin.
get the sea. Okay.
Excuse me, the sea cucumber.
Okay.
Thanks, Del. And the little warts and bumps that you're seeing, those are kind of like modified tube feet. So this is a deposit feeder. It's basically, its mouth is pointed down, it's ingesting chunks of sediment, and it will digest whatever organics it can find in there. This may be in the family Synallactidae. Thank you. A good view. We can move on.
Great. So we see these rocks outcropping here, this thick coat of manganese crust. We've been seeing that in a lot of our expeditions. It's one of the things we're trying to investigate on this project, this capstone project, distribution, the chemistry of this iron manganese crust. Potentially has some economic value for rare metals. And eventually here we'll try to get a rock sample at some point today. We find a good one. We'll be looking at the crust, and hopefully inside there we'll find some volcanic rocks that we can also analyze.
Should we be looking for rocks already? Or do we want to wait till there's a little
Let's see if we can get up a little further.
Yeah.
Yeah, it really looks like many of these have sort of slid down on top. I would be my guess, but I don't know for sure.
It's another of the Penaeid shrimp that we just imaged a little while ago. The direction that you're looking at, that we are moving in, is 247, 248 degrees, which means we're pointed kind of to the southwest as we move up this slope on the side of the seamount. We're at about 1,921 meters depth, so almost 2 kilometers depth, which is about 1.2 miles. Let's see a couple of crinoids. Pilot, to the almost directly left of the lasers, I see something brownish against a rock. No, it's in the foreground. It's below the lasers now. And I can't tell if it's another sea cucumber or if it's actually a sea pen. Now it's above the lasers. It's a little brown spot to the left of the lasers, about 10 o'clock, not too far from the rock that you're on. There's a smaller rock and there's something brownish just above it in the sand. Yeah, your lasers are almost on it. Yep. There you go. I guess now I see. I think it's going to be another of the sea cucumbers. Yeah, that's what it is. So we're just getting a slightly different view. It looks like the same species we just saw before, Synallactid. Now it's lifting its anterior end, and so you can see some of the that deeper pinker color. We've caught it with a mouthful there of sediments and just dropped some. Thank you. Another one of its cousins. There was a brittle star in the face of that rock there. It's another kind of echinoderm. 5 classes of echinoderm, and if you've been following along on our dives this week, you have seen them all in abundance. We have seen a lot of echinoderms. What's your gut feeling as you look at these rocks? Are these things that are in place and buried, or things
Yeah,
that
I
have fallen?
was trying to decide that. It's always a little hard to tell. My guess is there's some steeper topography up here. Many of these look like they're kind of sitting on top. They're kind of few and far between.
I do think I see some ripples now, kind of like moving across, right? We're
yeah,
parallel with
now I think, now maybe we just have a different view of it. But I agree. You can kind of see the alignment of that stuff that's fallen down, that pelagic material there.
Yeah, I can really see it on the right side of the screen. I can't really see it in the Sirius view. Oh, maybe a little bit.
Yeah, you can see it now in the Sirius view, yeah.
And this is Brisingid sea star that we're seeing, and it's next to a coral. So here's our first coral of the dive on the right side of the screen. If we can get a zoom on that, pilot. Thank you. So the Brisingid, the large arm thing, this is another kind of sea star. It's got a lot of arms, and it curls them up into the water column in order to capture its food particles from the water. So unlike a lot of other sea stars that are either eating in the sediments or preying on corals and on sponges and so on. So what will be our first coral of the day? And I think maybe there is something next to it that's interesting also. I'm wondering if that's an aplacophorine on the rock right next to it, or if it's the arm of something that's under the rock.
Could also be that weird hydrozoan, Scott, but
yeah.
I don't know if the depth's right. I think we saw it in the Marianas, if I'm not mistaken.
Right, right. You mean that that big solitary one? Oh, this has got a large...
Yeah, the one we collected.
Yep. Oh, this is interesting. It's got a large brittle star, ophiuroid, on its branches. You can see how fine the sediment is, so it gets stirred up by the ROV thrusters.
Please pardon the interruption. Your conference contains less than 3 participants at this time. If you would like to continue, press star 1 now, or the conference will be terminated.
Somebody on shore, please press star 1.
Saturday morning. I can't believe more people aren't called in. You're missing it.
So as we're turning around to this, I totally agree with you, Chris Kelly. I do believe that is a solitary hydroid, and I am currently forgetting the name that we thought we might be able to apply to that large polyp.
This may be a challenge for us here.
Yep.
With the
Pilot, if it helps, looking from the other angle is fine for the coral. Understood. This is actually, yeah, what the pilot's saying, this is actually a really good indication that there is a fairly strong current here because it's moving these particulates out, and when you're in places that have a quiescent flow, it just hangs there.
That's right. It is moving, and it's sort of moving off to the left of the screen there, which would be to the south here, I guess. And
so this is kind of, I guess,
kind
of a downslope current. So coral biologists on shore who are tuning in, this looks to me like it's a white plexaurid, which is a little bit unusual. I wonder if it's maybe a white paragorgiid. That would make more sense. Okay, copy that. So what have we got here? Well, so the yellow part that you're seeing is actually an overgrowth. That's a zoanthid that is overgrowing the Octa Coral, which is underneath. And the Octa Coral may be a coralliid, so one of the precious corals. And instead of being pink or red that we usually see, I believe this is a white morph. So that's my best guess, Coralliidae. And being overgrown by a yellow zoanthid, which is basically a colonial anemone. And you can see it's creating basically an entire sheet as it begins to overgrow this colony. And then it has a brittle star, astrosquamatid brittle star, in the center there. And then that really interesting pink
what
blob.
is that?
Yeah, so that's a solitary polyp. It's a hydrozoan, and I think when we zoom in, you'll see it's going to have lots of little tiny balls on the surface of that polyp body. A little bit hard to see. I'm not really seeing them. So those would have been small tentaculate-like structures, but I still think that's what it is, unless anyone's got a better guess. Anybody on shore? Perhaps nobody pushed star 1?
Definitely something new.
Yeah, so Scott, I don't know about whether there really is that same type of candelabrid hydrozoan or something. I'm not really sure what we're looking at here.
Yeah, thanks for reminding me of the name. Candelabrum is what the one was that we collected. I remember on the Johnson unit we also saw one of those. And this looks a little bit different, but it seems to have that same texture, and it's certainly holding itself onto the substrate. I mean, the only other thing it could be would be one of the what are referred to as the acoelimate worms. No problem. Excuse me. Unsegmented coelimate worms like the sipunculans and so on. So I do believe that's a solitary hydroid. Chris, any comment on the coral itself?
Well, if I heard you correctly, you were potentially calling it a coralliid. Is that right? I mean, you initially thought it was a plexaurid, and then you were moving maybe to coralliid. Did I hear you correctly?
Yes, you did. That was my best guess, having a look at it. And that would make sense with that ophiuroid as well.
Yeah, and I agree. I was going to coralliid just as you were saying that as well, once we got that good look at it, too. So, yeah, I think that's the best guess.
Great. So then we agree that it's in the family. It's likely in the family that includes all the precious pink corals. Those are corals that have historically been harvested for use in the jewelry industry. They have a very hard skeleton that can be polished quite nicely. I don't believe that there's been any harvesting for the white ones, certainly not at these depths. If we can get a quick look at this rock, I see a sponge and some crinoids and brittle stars. So let's have a look at the sponge in the back first, please. Since we have a self-proclaimed spongeophile on the phone.
That's right.
There's another plesiodimidemid urchin down there. Go ahead, Chris, if you want to walk us through this.
Yeah.
Sorry to put you on the spot.
Does that kind of say it all? I don't know what this is.
I don't think we've seen this sponge yet.
No, it's interesting because it's really thick-walled.
Right.
It's clearly has a vase-like morphology. There's no stalk that I can see to it, but it's so thick. That's what's kind of interesting. It's opaque. And in the foreground, there's a little tiny coral there. I'll bet that's a little bamboo coral. This has, let's see, 4 or 5 brittle stars. You can see their light pink arms with the spines. And then it's got a couple of crinoids as well. Both of those are different kinds of echinoderms. Sorry, Chris, were you trying to say something?
No, I might have had an open mic. Astrid asked me a question here. I honestly don't know what the sponge is. I can't even go to family at the moment.
Oh, that's kind of exciting. Is it interesting enough that you're thinking it's a possible collection target?
You might have been reading my mind there. I wasn't going to say anything, but I think this would be a pretty nice first specimen if you were able to do it, if you're in a position to collect it.
Yeah, let me check with the pilot. Pilot, do you believe that we are in a position that collection is possible? Okay. Yeah, I think it's probably connected on the other side of that rock. Okay, Chris, stand by. We'll also get a
sure,
good
and
view
it'd be good to get a look at how it was attached to. That, as you know, is very helpful in diagnosing what the species is.
Absolutely. There goes that Basilocetus cuskeel again, drifting by.
Yep, that looks like another bazo, along with a shrimp too.
I'm learning, Astrid. See another coral also on that rock in the distance, and this brings up this issue that we talked about fairly extensively on maybe dive 3, the difference between the sediments and the adjacent rock and how you get a completely different animal community on each of those substrates.
Yeah, I think that's something we've seen on all our dives, and maybe most spectacularly at the end of end of yesterday's dive.
Pilot, before collecting, if we could get a zoom in on where that sponge connects to the rock, that would be important taxonomic information. Thank you. Looks like that sponge is probably about 15 centimeters across at the widest part at the top.
So I'm not getting a really good look here, but I think it's almost fibrous where it's connecting.
Yep.
I don't know if that's your sense, but
Yes.
maybe we'll get a little closer look now.
I agree with that, Chris, and I was trying to remember that terminology that you were teaching us last year about the difference between the fibrous and more like the hold fast, but this does kind of look like the fibrous attachment.
Yeah, it's hard to say, but yeah, the terminology for a sponge that is attaching to the substrate with sort of these very elongate threads called spicules that sort of anchor it to the substrate. That's called lophophytous attachment. And then other types of sponges will actually glue themselves to rock with a nice sort of hard base. And it's a silicified base, and that's called basiphytous attachment. But this one, I honestly couldn't really say which way it was attached, just the way it's positioned there. But that's fine. If you're going to get a piece of this, then we should be able to figure out what it is, whether or not we see the base or not.
Thanks, Chris. So we're getting an interesting question from one of the Exploration Command Centers, IC Watch 2, and I believe that's the one in Rhode Island. Do you want to take our first crack at that, Del? And
Yeah, so one of the question here is about the amount of silts and debris that we're seeing here in the water column, and it seems like most of that is getting kicked up actually when we set the ROV down from its thrusters. But you can see it does move off pretty quickly, and so that's an indication that there's pretty strong current here to the from the right to the left along this ridge. So we've been talking a little bit already. We've got the sort of lighter sediment in the background, and we've got this material that might have come from the upper ocean and some kind of bloom event that seems to have fallen down on top of it. So that more organic rich material on top. And so you're seeing all of that kicked up, and it looks like the organic kind of material floats around a little bit longer. And of course those finer sediments settle out a little quicker.
And in addition to that is just the fact that we do see some of this material sitting on the bottom. It has to be drifting through the water column. So this may be a particular productive time of the year. So there may be a lot more marine snow that is drifting. But I would agree with you that we're probably responsible for a lot of what we're seeing kicked up so far.
So it looks like Tim would love to have one of these associates on the sponge.
Oh, I'm sure the associates are going to come for the ride.
Yeah. So if we can, going to try to grab it near the base here, I guess.
Yep. Yeah, it's not that large.
No.
It's only about 15 centimeters across the top, so I think we can get a good most of this sponge.
The lasers are about 10 centimeters across, so give you some sense of reference.
Historically, what we've seen when we're doing the collections, the ophiuroids probably will hang on to the sponge. The crinoids may swim off. Sometimes they do, sometimes they don't. So we'll see what happens with this.
Yeah. Oh.
So I see a comment from Emil Petruncio, who's adding to this discussion of the sediments and the organics that are on the sediments, and he's noting that we're far from any continent, and so we can be pretty confident that what we are seeing is settling out from the water column and isn't, for example, being carried off of the continent by rivers and that sort of thing. It's not like we're on the slope of an island.
Yeah, that's a great point. And we did see some material coming down from Jarvis, probably from the reef on their dive the other day. But yeah, here we're a long way from any other sources.
So I think this is going to be soft, but I could be mistaken. Just letting the pilot know there that it should sort of squish a bit as he grabs it, and then he'll tear it off. Hopefully that's the case.
Thanks, Chris. Pilot heard you. If you could try to grasp it near the base, that'd be great. Thank you. Yeah, we think it'll be a little bit flexible, so I think as you're moving the claw by it, it might actually push away from the rock a little bit. There's a good indication that it is a little bit soft. We saw it kind of move as the manipulator touched it.
Yeah, and that's informative. Him just touching it like that actually helps. So my best guess is this is either going to be in the family Euplectellidae or it's going to be in the family Rosellidae. Those would be my first two guesses. Perhaps in the family Rosellidae, because there are some of these vase-like sponges that occur in that family, most of which are not known very well.
Great, thank you, Chris. Copy that, pilot.
Yeah, and while we're waiting, in case we've got some new viewers who haven't been watching very many of these dives up until now, we've talked about hard sponges and we talked about soft sponges. So there are types of sponges that they essentially glue together their microscopic parts, which are called spicules. They glue them together and form a very rigid framework. And in some cases, this can be kind of a tough framework. And in other cases, it's very, very brittle. And if you go to take a piece of it, it's like one of these really thin cookies that kind of just shatters and breaks apart when you grab it. And then there's these other sponges that don't glue their spicules together. They're kind of loose in the tissue, and those are your soft sponges. And that's what we think this one is. So when he grabs it, it's going to actually, if you were feeling it with a real hand, it would feel like your kitchen sponge to a certain extent. It would kind of squish, and then you'll have to sort of tear it a bit to get it off of the base. But that's the main difference, and just knowing that one fact about a sponge can eliminate a lot of candidates.
Thank you, Chris. So we are at 1,912 meters depth. We are on Keliʻihananui seamount. That's the proposed name for this seamount. This is our first collection. It is a glass sponge, possibly in the family Rosellidae. You can see that it has some brittle stars on it. And there's a couple of crinoids. We'll see what they decide. Looks like one brittle star is deciding
bail, yeah.
He does not want to stay with the sponge.
Oh yeah, broke off pretty neat there,
it's
didn't
pretty
it?
soft. Pretty soft, like Chris said. Yep. It depends. So I'd like to see the bigger piece. If it just depends how confident. I mean, there is an ophiuroid in there. If it's easy to drop in the box, let's take it. Okay, let's take it. Thank you. Because I see both a crinoid and an ophiuroid in your claw right now.
A little bit of everything there.
Yep. So that way we know we have something.
Looks like we're going to inside port.
Yep, and I see it tumbling down there. So we know that we definitely have a piece of that sponge along with an ophiuroid and a crinoid, and we'll now see if we can get a bigger piece of that sponge to put into the box. But we definitely have some, so that's great. Tim Shank's asking this really interesting question. He wonders if these vase sponges fill up with organic detrital material after repeated surface blooms. And that's kind of interesting because it's not unusual that we see these dead sponges. They're much more brown in color, and you wonder, well, what killed them? And so it's...
yeah, sediment goes in, doesn't look like it's anywhere to come out there.
Exactly. Now, I mean, the one thing is that the direction of flow, these are what are called aquiferous animals. They have basically a plumbing system, and they have cells that generate a current that moves from the outside to the inside, and then it flows out that
Might be able
vase.
to push some of it back out then.
Right, exactly. So I guess it depends how rapidly is the material settling inside. That's a really interesting question, Tim.
I was wondering too, Scott, before we leave here, after he grabs the piece or before, if they could do a zoom on that base and attachment now that we could actually see it more clearly.
Copy that, Chris.
So Emil is just commenting here. He's been doing a little research on these sediments out here in the Central Pacific, and he's commenting. It looks like most of this sediment-defined stuff we're seeing is the siliceous ooze, which accumulates at a rate of about a few centimeters per millennium. All right, so he says we're kicking up lots of history here.
Yeah, that's it's a really neat way to think about it. You mentioned earlier the bioturbators, the animals that are ingesting the sediments and digging through the sediments, and they're also influencing history by kicking up some of the older stuff and putting it on the top and so on. That's a really cool way to think about this. So siliceous material settling, biology, that would be things like diatoms. These are single-celled phytoplankton that build a shell that's made of silica, and also radiolarians.
Yeah.
Those are the two obvious ones to my mind. So delicate. It's like picking up bath foam.
Said than done. Or
Sorry. like one of those crinoids just bailed before going into BioBox, but the other one is still hanging on, and very nice.
There we
Wonderful.
go.
Nice job, pilot. That really saw sponge. You can see there's a little piece of it drifting by there, but the bulk of it went into the BioBox, so we have a really successful collection. That second piece that was picked up also had a crinoid attached to it, so I believe we got 2 crinoids, 1 or 2 ophiuroids, and the sponge there. Nice job. Before we leave, pilot, let me know when you're settled. There's a couple of close-ups on the rock we'd like to get. Great, thank you. So the first thing we'd like to see is the base of the sponge that we just collected. We're trying to get an idea of how it actually attached to the rock. The second thing, just to let you know in advance, will be at the bottom right side of the rock. We can't see it right now. There's a red brittle star. Sure looks to me, Chris, like it's going to be the Lophophytus attachment.
Yeah, it's a little hard to see. I think.
Yeah, we're just getting we're getting into position, and we'll get a good close up here. No, I think unfortunately the attachment is on the other side of the rock.
Yeah, that's okay. I think we probably have enough to figure it out, and also given the piece as well. So you might as well see if we can get Tim's brittle star there, wherever that was. I think it's the one that was a little bit below the ROV now.
Yep, so if we just back up a little bit, we should see coming into view a red brittle star on the lower edge of this rock. You're probably actually in a really good position to see it. There it is, yep. All right, Tim, if you would also like to say something on the phone about this.
I think Tim might have stepped out.
Oh, okay.
Not sure if he's back yet or not.
Well, we'll get him a quick close-up here so we can look at the imagery later.
I just want to say thank you for that sponge. All the other sponge people, I think, will be very grateful, because that looks like it could be another potential new species, most definitely a new record, but very likely a new species too.
Fantastic. That's great stuff. So I would say that this is an Ophiacanthid brittle star, and that's a family name. And the main character that I'm using to suggest that is the long spines that are seen on the arm. But Tim was also saying something the other day when we did zooms that it had to do also with the size of these, the different units of the arm, what are called the vertebral ossicles. You can see those appear like rings. Very round disc. Interesting that the tips of the arms are all curled. It's kind of an interesting pattern that the spines are making when they're curled there. I don't know exactly if that's some way to enhance capturing something.
Well,
no idea.
I think you can certainly sense all the sediment raining down. Probably maybe some reaction to that. I don't know if he was like that when we first came in or not.
It's cool. All right, thank you, pilot. I don't think we need to do that. I think we can make some headway. So you can see on that, excuse me, on the far left, that's a Brisingid asteroid, another kind of sea star. And then where the lasers are, we're just looking at a close-up of an ophiuroid brittle star, and then to its right is a crinoid. So 3 of the classes of echinoderms right there in a
One
row.
rock.
So what are we missing? We saw the holothurian, and no, we saw the urchin.
We saw the
So we've
urchin,
seen all 5.
Sea cucumber, yep. Cucumber and urchin.
Yep. Sea cucumbers, sea urchins, sea stars, brittle stars, and feather stars or crinoids and sea lilies. That's great.
All right. Still think we need a name for that when we see all 5. I don't know.
I still like the royal echinoderm
Royal
flush.
echinoderm flush? All right.
Sure.
Let's go with that then.
There is a whip-like structure just coming up on your right side. If we can get a quick zoom of that. Let's see, we are seeing a coral almost on its perpendicular side. It looks like this is a primnoid Octa Coral. It's the family Primnoidae. Once again, it has one of these, what are referred to as astroschematid. That's the family brittle stars in its arms. I also see right on the tip, there's a little mysid shrimp. Those are called possum shrimp. You can see it's got a very thin tail and kind of an orange body. So we're looking at this on edge. It's kind of a fan.
Mm-hmm.
But I can recognize the structure of the polyps even from these distance as being in the family of Primnoidae. And we'll see if any of our onshore scientists have a better identification than that. And there we see one of these urchins in the background. So this
Scott, I would definitely agree with you that it's a primnoid, and it seems to me that it's got more or less plumate branching. I don't know if you saw that also.
Yep, exactly.
Which could indicate that it might be something like Calogorgia, but I'm not certain.
That's exactly what I was thinking, Chris. It looked like a couple of feathers. The branches were in a feather-like appearance. Chris described that as plumose. And that is a characteristic of the genus Calogorgia. So that is a possibility. What we really need to do in order to identify these primnoids is look at details of the polyps themselves, but sometimes the overall branching gives us a good indication, and that's a good view that we're looking at right now. You can see what we mean by this kind of feather-like branching appearance.
I'm guessing this has a holdfast on the rock here. It's just a little bit of sediment sort of covering it up.
Absolutely, good observation. So this larva would have to settle on the rock, attached to the rock by a structure called the holdfast, which is at the bottom of the skeleton. And you can see your mysid shrimp there again. It's 2 big red eyes and the orange body and the little abdominal tail sticking up. Good view of the ophiuroid. Sometimes the characteristics of that central disc are used to identify it, so I think Tim Shank will be happy with this view. Nice stable view. That's great, pilot. When video's happy, we're happy. And I noticed as you were zooming in the background right, there's another colony on another rock. That will be different. Yeah, it's really small, so I don't want to spend a lot of time getting in position for it, but if we get a snap zoom maybe. Thank you. Yep, it's exactly. It looks like one stalk. It's a whip. It's whitish. Looks like a little bamboo coral from here. Nope, it's another primnoid. So this is in the same family, the Primnoidae, and right now it's unbranched, and there is a genus Candidella that has some unbranched primnoids in it that have very large polyps, and it goes by the name Candidella gigantea, so I suspect that's something related. Thank you. It's funny, a couple of dives ago I was remarking that we're not seeing any primnoids, and it's a really common family in the deep sea. It's got a lot of different species. So our first 2 or 3 today are primnoids. That's a shrimp sitting on the bottom there. Yeah.
Yeah.
So, Del, remind me, I believe we were kind of targeting being in that potential mass wasting area, right? The possible landslide area.
Yeah, over on the kind of southwestern side, it looked like there was potentially there's a big slide block, some steeper topography, and then a kind of a mass of stuff down by the slope. And so we edged over just a little bit to try to catch that. It's difficult to see that we're seeing much that would be diagnostic of a landslide or a mass wasting event at this scale, but we're kind of keep our eyes open here. Certainly looks like the surface has been here a while. It's pretty heavily draped with sediment, and we've got really thick manganese crust. So if we are on the edge of that mass wasting event, maybe that happened a long time ago. Go ahead. All right, so we're just doing a little pilot shuffle here for lunch. So let's give us a few minutes, and we'll be back in, and we'll keep heading up slope. Yeah, we go ahead and take a zoom in here if you want. Yeah, why don't we do a quick zoom here? I think there's a rock here with a crinoid, and yeah, just do a quick check out here before we head out. Yeah, so we see a couple of crinoids here, and then
Sorry, that.
But if we could get a look at that red coral in particular, that crinoid right in front of it, we're sort of guessing it's in the family Chiritometridae, and it could be in the genus Glyptometra. Okay, so this is a Paragorgia. It's a bubblegum coral. Presumably it's got that nickname because of those balls at the end of the branches that are quite obvious when they close up their polyps. So all the polyps on this colony have closed up. And then these yellow polyps that are open, these are larger zoanthids that are overgrowing the Octa Coral colony itself. So that's a really, really beautiful view of a Paragorgia. I'm not sure we can go any further than that on the identification. If Santiago Herrera was here, he is a specialist in this group of corals. He could probably help out, but I certainly can't.
Great. Thanks, Chris. Yeah, that's a great shot. Yeah.
So this is a great shot of how this crinoid has got himself on the substrate here. Those little almost root-like structures, as though you were dealing with a mangrove tree or something like that, those are called cirri. And the way in which, how long they are and whether they can curl up multiple times, like prehensile cirri would be ones that the crinoids or feather stars use to attach the corals because they can wrap them around multiple wraps on a branch. This guy does not have prehensile cirri, and that really helps with the diagnosis. So Chuck Messing, I don't know if he's watching, but my guess here would be in the family, as I said, Chiritometridae and possibly Glyptometra, the genus. But fantastic shot of that, and Chuck, the expert on this group of animals as well, he ought to be able to get a really solid ID from that imagery.
Great, thanks, Chris. Yeah, it didn't look like he has much to hang on to there on that manganese-encrusted rock, but that was a fabulous shot. All right, so we just keep making our way up the slope here. We're seeing a lot more rock than we have in some places. Looks to be fairly crusted in. Yeah, that's a sea spider. We saw one already. Let's just a quick zoom if that's another sea spider. I can't. Okay. Oh yeah, there I see it. Perfect. All right. No, I think that's good. We've seen this already.
Right below the ROV, there was a carnivorous tunicate as well, that really clear gelatinous
right
thing
there,
that was on the rock ledge.
Yeah, let's get a zoom on that if we could, please. So yeah, what are we looking at here, Chris?
So this is a tunicate, and it's a sea squirt, but he's a very unusual sea squirt. At least you don't see them in shallow water. You do find them down here, so they're not that uncommon, but they're really unusual. And they're always sort of transparent. His base is sort of coming off to the lower left of the main body of it. You can kind of see this gelatinous part that continues on beyond the round part, and that's what he's attaching to on the rock. And then he's waiting for some poor, unfortunate invertebrate to swim by, I gather, and go in one of those openings, and he has a way of trapping and eating them, and I don't understand how that all works. But this tunicate is presumably in the family Octanemidae, and people go a little further than that, but I don't. So carnivorous tunicate, nice shot.
Wow, that's an amazing looking animal. I've never seen that before. Uh, Yeah, we have not seen the stock crinoid today. I think this is our first stock crinoid, so let's zoom in here.
So Chris, this is a really bright red crinoid. Would you mind talking a little bit about why, when we see organisms like this in the deep sea, why would they be such a bright red?
Yeah, I can make some stuff up, I guess, because I don't really understand it myself. But you obviously don't have any red light frequencies at this depth, except from other animals, perhaps that are producing their own light through bioluminescence or something. But in any case, red is going to be black at these depths. There's, a lot of the I don't think that many things actually eat crinoids, and that's why I think they're fairly successful. So why this guy is red, I don't really understand. But it sure helps us out in identifying them, because this is a very, very distinctive sea lily, which is the common name for these stock crinoids. And it's called Proisocrinus ruberimus, and the color is one feature, the number of arms is another feature, and then right underneath the arms themselves where the calyx is, you can see a number of sort of reduced cirri on it, and also this foot, how it's attaching is very distinctive. But underneath the calyx that we saw earlier on, it's got these little projections or cirri, and that's also diagnostic. So it's one of the few sea lilies down here that I think we're fairly comfortable going right to species, and they're just magnificent. I love to see these things, no matter whether it's new or unusual or not.
Thank you, Chris.
All right, pilot, I think we can continue.
I guess since we're looking at them and moving on, one last comment about stock crinoids, again, for people who are new to this broadcast. These are considered living fossils. Their morphology has changed very little over 100 million years or more. They're a very common animal in the fossil record. They've got a lot of hard parts that are preserved. And so crinoid fossils are everywhere in these, and that particular one would look almost identical to a number of the ones that are fossilized in rocks that you could see at in museums. So they're really, really fascinating. They evolved, if you will, at a very, very early period, and they've remained essentially unchanged for an extremely long period of time. So that design obviously is very successful.
Yeah, it certainly, as a geologist, that's when I think of a crinoid, that's what I think of from our paleontology classes. I can remember being a kid and picking these things out of limestones in central Illinois that were 300-400 million years old. So, like you said, a long history, and must be a robust design. Yeah, so our pilot Jeff here just commenting that red is absorbed more quickly in water than other colors. So, Oh, here we go. Let's see what we got. I think it's a sea cucumber, yeah. We saw a red one earlier. Maybe someone on shore can offer us some assistance here. Oh, purple Heratherin, Jim is saying.
Well, I'll take a stab at this one. Could be a couple different things, but my best guess is something, well, it's in the family Synallactidae, I believe. And this guy will swim if disturbed, I'm pretty certain. And this could be in the genus Paleopatides, just from the coloration and general morphology, but I'm not really certain. I don't have a good grip on the sea cucumbers, even though I've certainly tried to learn something about them. But very, very beautiful sea cucumber, and over in the Maui Ocean Center, which is showing the live feeds, they actually were calling this thing the purple people eater, one we saw a couple of years ago. So we could give that as a nickname.
Tim, are you on the phone? You want to chime in here? I'm not sure if you're on the line or not. Yeah, a few people are agreeing with you here in the chat room, Chris. This guy would be a swimmer. So we didn't disturb him enough, but
yeah, it's interesting to see that there's no real trail behind that one. There's no Lebensspur, and there's no remnants of detritus nut guy. You know, no fecal casts or anything, and no trail. So maybe that it sort of landed there and has been hanging out and ready to take off and swim again soon.
Yeah, that's a great observation. We saw a lot of those big casts or trails when we first sat down. We haven't actually seen one in a while. Yeah, we can just take a quick look at this. I'm not... From this angle, it's hard to see.
Yeah, so this is this same Gorgonian we imaged a little while ago that Scott had identified as in the family Primnoideae. And we thought that perhaps it could even be in the Caligorgia genus, a genus in the Primnoideae. This particular genus tends to have smaller polyps, and it has a plumate branching structure, so that's what we're looking at. They look like feathers, each of these branches. And one of the distinctive things that you look for within this genus, if that's what it is whether the branches or the plumes on either side of the branches are opposite each other or alternating. That was alternating. And so that's informative. I only know of one alternating species. There's probably a lot of them, so I won't go further than possible Caligorgia. But anyway, beautiful primnoid Octa Coral.
Coming up on another big exposure of rock here. This has traditionally been where we've seen lots of interesting stuff. We got a, looks like a crinoid there on the right. We've seen plenty of those. Yeah, I want to do a zoom on that there. Well,
Okay, well, I guess I'm one of the vocal biologists here today since it's Saturday and I can actually spend some time. So this is a feather star. It's a unstalked crinoid. And so these guys aren't attached via long stalk. They just sit on the bottom and they hang on via those cirri. And we've seen a couple of these, the yellow one, the Glyptometra. The distinctive thing about this one is it has 5 arms on it. The other yellow one had a lot more arms, but 5 arms is potentially diagnostic, and at least for the moment, I think we could go with possible pentametrocrinus. The name actually implies that it has 5 arms, if I understand the root correctly.
great. Thanks, Chris. Yeah, we've seen a lot of these yellow or green kind of non-stock crinoids. I think this is the first one we've seen. It's this color here this morning. All right. Yeah, that should be good, Pilot, whenever science is ready, whenever. You zoom in there, you can really see the crust forming on these rocks. Manganese crust.
So, Dill, are all these little pillow formations here, or are these just kind of cobbles that are severely manganese crusted, and that's why they're so round?
Yeah, that's a great question. That's something Deb and I were discussing the last few dives as well. We get a view here. I think the overall morphology is that of the lava, but certainly the finer texture you're seeing, these little knobby patterns we see on the rock, that is from the crust. But the overall structure of this thing, it looks like a volcanic ridge to me. Yeah, it's looking more and more as we go upslope here that these actually are intact rocks that are outcropping here, and the higher parts of the ridge, we're seeing the sediment sort of swept off them. That's why we get this exposure. We really need to have these rocks exposed over long periods of time to develop these thick crusts. So let's do a quick zoom here. I think we've let's do the star. I think we've seen plenty of those yellow crinoids. See if we can get a shot of the disc here.
So this is kind of a cool shot. Hopefully Chris Ma will call in. But it doesn't just have 6 arms. It actually has 11 arms. But those upper 5 ones, they ate them or it lost them somehow, and they're just starting to regrow. Really interesting observation on this guy. And we'll find out if Chris is on the line. And if not, then I'll go ahead and try an ID a little bit later on.
Thanks, Chris. I'm back.
I'm going to venture a guess. Maybe some fish predators came and ate those arms.
Thanks, Astrid. Just wanted to let you know I'm all back from. Let you all know that I am back from lunch. Maybe not all of me, but yeah, it looks like there's been some predation and regrowth here. Thanks, Pilot. So we are at 1,877 meters climbing the slope. I see there's more rock exposed, and when I went to lunch, you could see a pretty good slope here to the bottom of this feature that we're climbing today. Have you guys in the front row had an opportunity to introduce yourself this morning? Please do so. I'm passing over another of the synallactid sea cucumbers. Go ahead.
Yeah, the front row here we have at Pilot is Jeffrey Lanning. The rest of you guys want to introduce yourselves?
As co-pilot, Levi Unima.
As navigator, Fernando Aragon.
And in the back row?
Annie White, in the clipping chair.
And here in the front, you can't see, this is Roland Bryan, video. We're all working for the Global Foundation for Ocean Exploration. There's a fish floating upper right.
We'll see if we can get a, looks like this fish is kind of head up. Oh, it's gotten itself into a horizontal position. Yep, just to be different.
I don't know if it's different. It looks like another bazo to me.
Okay, another Basocetus.
So that is a basocetus, which is in the cusk eels, the Ophidiidae. Seems like this is one of the most common things down here in terms of fish species. But he does have a long pectoral fins, it looks like. A nice lateral shot would be good.
Not sure that that's going to happen. There we go. It looks like today's going to be a day I say something, we're going to get the opposite.
So what Chris and I are looking at here is the pectoral fins to try to see if it has an elongated lower section to those pectorals, which would make it actually not a basocetus and maybe an eretmixes. If he cooperates, maybe we'll be able to make that call.
I do not see an elongated lower part to the pectoral fin, if that helps you.
All right, so I think we've gotten a good view, and we think basocetus was on the money before. I don't see that elongated lower section there.
Thank you. We can move on, pilot. I noticed as we were following this fish and moving over the bottom, there's several of the plesiodiademid urchins, the ones that are on the long curved spines, and some of them were moving at a pretty good pace. A couple were being, I think, moved by the ROV current, but something interesting. Yeah, it is interesting because I don't know what it is yet. Huh. So possibilities are a sea slug. Actually, I think it is a sea slug. Yep. So it's a different morphology than I've seen before, but I think I can see the head just sticking out a little antennae maybe there at the lower left of what we're seeing, this view. Yeah, I wonder if this is another Sacaglossan type. We saw one of these, I don't remember if it was yesterday or the day before. Another suggestion is that it's a sea cucumber holothurian, but I don't know, something says to me that this is a mollusk related to a snail. Doesn't seem to be moving at all. Yeah.
So there's some projections, white projections coming out of the bottom of it. Do you see those, Scott?
I do, and I was thinking that those may be sensory antenna and not tube feet, which is why I suggested that it could be a slug of some kind. But I do recognize that another possibility is that it is a sea cucumber, just a highly modified sea cucumber. What do you think, Chris?
Well, I was at first going to sea cucumber, but now looking at those projections, I think you're right. I think this is some type of a pluribranchid, one of these very large deepwater sea slugs, but I've never seen one that looks like this before.
Yeah, so the other thing I'm looking at is you see the way the front margin seems to be curled around and is leaving an opening there that looks like there's two lobes on either side. So that doesn't say holothurian to me. That says something more like a sea slug. So that'll be interesting. We'll send that image around. Thank you, pilot. I think we can have a move. I mean, unless that thing were to open up, we're not going to really learn more about it. I mean, that kind of looks like that little opening, like there's a siphon in there, which would further back up the fact that it's a gastropod mollusk and not a sea cucumber. I had a note here that we did see a bubblegum coral, but let's get a quick zoom, and that does look like what this is. Yeah, so you can see it's very fuzzy where the polyps, the feeding part, the tentacles are being extended from what we call the cenonchyme. That would be the more red material you see, which is the tissue that connects all the polyps and houses the skeleton. So this does look like a bubblegum coral, and it has an ophiuroid brittle star and its branches, probably the family Astroscomatidae, and on the lower left side I saw there was some yellow, which suggests that it also has a zoantid overgrowing it, and I think that may have been the case for the last one you saw, according to the notes that I see here that Dell left. Thank you. It's a good view. And again, make this note that that coral is growing on that rock, and that's not an accident. The larva wouldn't do well if it had landed in the settlement and tried growing. It needs a stable attachment in order to grow upwards and then be stable enough that when the currents are passing over, it doesn't get knocked over. Those polyps are picking particulates out of the water column, so they need to grow up into some kind of a current. I see a few scattered brittle stars on the rocks here, and you can see some more evidence of the rippling. It's kind of perpendicular to the way we're moving. Or excuse me, we are parallel to it. So the currents would be perpendicular to our view, moving from one side of the screen to the other to create those ripples. It looks like we're getting a little bit more steep here. A lot more exposed rock. I will take a chance. I'm not sure. Let's see. We don't have Deb here. Dell is right now at lunch, but I'm wondering if these are little pillow formations, evidence of long ago eruptions that have been now coated in, or encrusted in manganese. So the colony on the upper left, if we could have a zoom on that, that may be. Yes, please. The white looks like there's no branching. I'm thinking this might be a bamboo coral, but the last one we saw like this that we zoomed in on was another primnoid. Yeah, so this one's a bamboo coral. I don't know that we've seen any bamboo corals today, so this would be our first. And one of the ways we can tell that is by the bright white of the skeleton, the thickness, and then you can see through the tissue there are some bands, especially at the lower part of the colony there where there were no polyps. I saw 2 or 3 of those bands. There you go. Those are called nodes. They're made of protein. And then in between the white you see is a calcium carbonate skeleton. That's called an internode. And one of the things we use to identify these is whether or not they branch, and do the branches arise from the nodes and the internodes. In this case, it's either, if we can get a zoom, just those lower two polyps, I see something sticking out to the left, and I wonder if that's a new branch that's forming. Yep. But that would also let us look at these polyps. So one of the things you can see, there appears to be needles that are extending between each of the bases of the tentacles. That's another important characteristic that we use to separate out the different species. So one of the questions, this is a relatively small colony, so it may be a very early growth stage of something that will branch later on, or it may be something that remains as a whip. And so it's hard to say. So the actual identification, you need to actually look at the polyps and the structure of those needle-like sclerites and other things that are in the tissue. Huh. Yeah, it's a little hard to tell if that's a branch. It's discolored. And so it may be that there was something that settled on this and has been overgrown by the tissue. It may be that it's a new polyp, but I'm just surprised because of the difference in color. But while we're zoomed in here, so you can see these 8 tentacles. That's a hallmark of the Octa Coralia. And they have little side branches. Those are the pinnules, and you can see there are little needles, pretty prominent needles actually, between the tentacles. Those are sclerites. And sclerites are also microskeletal elements, and so we saw that bubblegum coral a little while ago. It would have a very different kind of sclerite. They would all be glued together, and that's what would form its skeleton. I guess there's a little crab in there. I thought I saw something. A little squat lobster, yep. Very interesting. The kind of peachy orange color that you're seeing through the polyp, that would basically be the pharynx, or the throat. That's the upper part of the mouth. And it looks a little bit frilly right at the base of that, and then it's kind of hollow underneath, and that would be the stomach space. It's called the gastrovascular cavity. And each of these polyps are connected so that they're essentially sharing their stomach. So if one of them catches something, they can share nutrients. Thank you. Wonderful view. Great. There's something fuzzy and orange just above this on the rock. Copy that. You can see the holdfast, which is the sort of the disc that holds this colony to the rock, is really quite small. It's not too much larger than the skeleton itself. And this is a relatively robust skeleton for the size of the coral that we're seeing. There it is. Very good. That is a full holothurian. So here we're looking at a sea cucumber. This is a different species than the one we were seeing before, and it's so transparent that you can see all of its internal organs, and in particular, you can see its intestine. These holothurians, these sea cucumbers, don't really have a stomach. Instead, they just have a very, very long intestine from shortly after the mouth to the other end, and this thing is just packed with sediment. So you're seeing all this sort of yellowy brown material are sediments that are being processed, and the organics are being digested out of that sediment. And so you can see it's very highly coiled, and very early in the dive we saw what I said was a fecal trace, and it was highly coiled, and I said that was an evidence of a certain kind of holothurian, and that would be one like this. Great view, thank you. The feeding end is in the foreground down below, and I can kind of see these modified tube feet that are pressed against the rock. They can use those tube feet almost like little hands and scoop up bits of sediment and sort of cram it into its mouth. Les Watling, who's biologist at the University of Hawaii and is an expert in the bamboo corals and the chrysogorgid corals, has indicated that that bamboo we were looking at may have been in the bee clade, and we talked a little bit earlier in the dive about how we understand the relationship. Oh, there's a Metallogorgia on the left and another bubblegum on the right, so let's go to the left because that'll be the first for the dive. Sorry, so when we say bee clade, what we mean is we've done some genetic analysis, and there's a cluster of corals that are all related to each other that don't yet have a name. They're all in the bamboo coral group, so for temporary purposes we just refer to them as the bee clade. Soon that will have a genus name. And Les Watling also passed on that was a fantastic shot. We could learn a lot about the identification just from those wonderful images from the video. So this is in the family Chrysogorgiidae. We've been seeing these on a couple of the dives, not all the dives, but a couple. They're very characteristic at this depth. And you can see they have this really nice growth morphology, this long central stem or stalk, and then at the top all this branching, and right in the center of the branch is a brittle star. Very characteristic. In fact, we know that this is Ophiocrius, and I'm just forgetting the name of the species right now. That was the genus name, but we always see one of these brittle stars associated with this particular kind of coral. Very tight association. We never see that brittle star anywhere else, so we believe that the larva probably settles on the coral when it's very young. There's a very, very thin sheet of tissue. You can actually see the skeleton through the tissue. It's a little bit dark, and so you can see how thin the tissue is on that skeleton, and the polyps are almost just kind of like sitting on top. Thank you, that's a really nice view. In behind, I see another sea cucumber on the rock in the background, several brittle stars, and perhaps a sea star that we haven't yet seen in the lower right corner. Yes, please. There's also a little mysid, the possum shrimp that was swimming around on that Metallogorgia colony we just imaged. And I see in the foreground another one of the Plesiodiadema urchins. Yeah, there's a tiny purple colony tucked down there in the rocks, isn't there? Almost in direct line. Yeah, if that's we get that first. Thanks, Tim Shank. He's reminding me of the species name. It's Ophiocrius oedipus. That's the species of that brittle star. So we have a little Octa Coral tucked in down here in the rock, and that looks to me like one of the stoloniferous forms. It grows as a mat or a ribbon. And the ones that we know anyway are called Clavularia, so that may be in the genus Clavularia. Thank you. So those wouldn't create an upright skeleton. They would just continue to grow as a mat on the rock. Oh, and there's a squat lobster down below it. So now we're going to have a look at the sea star. So Chris Kelly, you are our echinoderm expert for the time being. This looks different from anything I've seen so far on this leg.
Yeah, we have some visitors here, Scott, in the ECC. I wasn't watching, and then Acid told me I was being called to identify a sea star that I don't know what it is. So we're going to have to wait for Chris Maw. I thought I knew what it was. I think it's something that may show up in the guide next year, but I don't have my notes yet with me right now today.
Okay, thanks, Chris. It almost looks transparent, kind of interesting. If you can get it, thanks. So that was Chris Kelly, I heard. He was at the University of Hawaii, and Chris is the science director for the Capstone expedition, which is the 3-year expedition that the Okeanos Explorer has been engaged in. This is the 3rd year. And so Chris has been looking at a lot of these videos. He's been seeing all of the dives that have occurred to now in this last 3 years, but he's also been working at Hurl for many years, and so has had experience with submersible dives around Hawaii and the Northwest Hawaiian Ridge, and so he's seen a lot of the animals down here, and they've prepared a guide. So if Chris says it's something a little bit unusual and new, it usually is. So we have seen, let's see, a Metallogorgia, Seaward and a bamboo coral. We got some beautiful images on. Another one of the bubblegum corals. 2 actually. There's another bubblegum coral right over here. We're seeing a sea star tucked in. Yeah. So I know we've seen some other sea stars that get tucked into these rocks in this position. I don't think this is the same as those, but that's a nice view. You can also see the red dots right at the end, right at the tip of each of the arms. And I think that's enough. I think that's good. Yeah, we can move on. Thank you. Those red dots are usually sensory tube feet. They have a pigment associated with them. So if there were light, they could detect it. I don't know what they would use it for down here. I think we're going over a couple more of those Caligorgia primnoids on the left side. We imaged those before. This yellow one maybe is different. It might be a bubblegum coral that's completely overgrown with zoanthids, but maybe just a snap zoom to verify that. I suspect that's what it's going to be. And I'm saying that based on the lower part of the skeleton. There is another kind of sea fan that we see that's yellow, but it doesn't have a base like that. And yet I'm wrong. So this is that other kind of yellow sea fan. So these are all Octa Coral polyps. This has not been overgrown. And so this may be in the genus Parapodia. It's in the classical sea fan group of Gorgonian Octa Corals. And it also has an associated ophiuroid. So we see this really commonly. There are a lot of different kinds of these brittle stars, and many of them associate with corals, sometimes different corals. And you can see lower on in the skeleton there's tissue been removed, but it does seem to be recovering. I see some small polyps trying to arise there. So good stuff. Another new coral for our day.
So are these brittle stars coraliferous, or is that right?
Well, right. So the general thought is no. They are not feeding on the coral. Thank you, Pilot. We're good when video is good. They're not feeding on the coral. They may be feeding on mucus that the coral is sloughing off, or they may be settling on recruits that or other organisms that are landing on the coral, and that would be one of the benefits and why the coral would benefit from having it there, is that it's keeping it clean, basically. Looks like a large vase sponge coming up here on the left. We'll get a quick look at that. Oh, I see you're behind, so just a quick look if you can. Understood.
Okay, I'm on it. As soon as you zoom in, I'm ready.
All right. You can start right now, Chris. I don't know how long we'll be able to linger on it.
Well, I can take a shot already. I'm pretty sure this is a euplectellid vase, a glass sponge in the family Euplectellidae. And I think this one is in the genus Regadrella. It's got a lot of the very typical characteristics for that genus, including all the little holes on the side, the osculum, and over the main osculum on the top, you see what's called the sieve. That's that spider web type structure. It's a covering, perhaps to keep certain animals from going inside the sponge. And this is a beautiful example, extremely healthy version of this type of sponge. It attaches by gluing itself to the rocks. And I don't know what else to say about it at this point, except for marvelous imagery.
Yeah, it's great imagery, and you can see all around that margin at the top the tips of some of these spicules that you've been referring to a few times. These are the siliceous structures that the sponge uses to create this architecture. And so you can see them almost looking like fine needles at the top.
Wow.
Yeah, really cool stuff. So again, this is a plumbing-based organism that doesn't have a stomach and a mouth per se. I just saw the claw of a crab being retracted, so there's a crab living on the inside of that sponge. So, right, there's no digestive system per se, as we would think about a digestive system. Instead, each of the cells has to pick up its own food. So really, it's only feeding on stuff that's small enough to be engulfed by a single cell. But some of those cells, they're called choanocyte cells or collar cells, have flagella associated with them that start the current. And so the millions of those cells together have a pretty good current. They get sucked in from the outside. Small particles are pulled out and ingested by each cell, and then the remaining water flows out through the top of that sieve plate that Chris was noting. Thank you. We're good when video is good. Beautiful image.
Have we seen smaller versions of this on this expedition
Well,
already?
so we
similar?
definitely saw that genus a couple of times on, let's see, what dive was it? I have to go back to my notes here. It may have been Kahalavai, but I think it was actually the ridge before that. Yeah, it was. It was at the Cook Islands, so it was when we were on the ridge in the Cook Islands.
Okay.
There was a lot of interest, and we saw at least 2 different morphs, I think, of that Regadrella. There's another Metallogorgia colony growing off the rock on the right side. We're just passing it. You can see a very long stem and then the massive branches at the top. We just had a nice image of that earlier. And kind of like the Chrysogorgia, every time you see a Metallogorgia, you can be confident to say that there will be an Ophiocreus oedipus in its branches, all right? So here we have aligned next to each other, a sea star, the pinkish one on the left, and then several crinoids to its right, both kinds of echinoderms. Yep, I think this is the first Anthomastus of the day, so just get a quick look at this. We really know what Anthomastus is. There are actually a couple of, well, there's several different species, so we probably can't identify it to species, but at least to just document that it's here, it's a good idea to get a close-up.
So this is more for the pilot, Scott, but we just had some visitors here that are attending some type of meeting on engineering, and they're to a NASA type of a conference, and they were fascinated by the technology here. We were busily explaining how this whole process of telepresence worked, and they were asking about the ROV camera and a variety of things, so they were really enjoying the show that the Okeanos is putting on right now.
Thanks for that, Chris. I believe the pilots heard you. There might be a little apocopheran worm just down on the, like about 4 o'clock on the rock underneath this Anthomastus on the right side. And that's a little worm-shaped molluscan predator of corals, so this Anthomastus better watch out. All right, thank you. There's actually 2 Anthomastus next to each other. The one on the left is a lot younger than the one on the right, as evidenced by the number of polyps. Yes, please. I think it's actually a cup coral. But you're right. It's a shell. Let's see. Oh, it's actually a living snail. Very nice. So that large tube that you're seeing coming off the right side, that's the siphon. So that's what it uses to draw the water in. It can kind of sniff around its environment and maybe head towards something that might be interesting to eat. Many of these snails are predators. Some of them are grazers of material that would be growing on the rocks, bacteria and so on. You can see one sensory antenna just sticking out there to the right side in front of the siphon. And then you can see that white muscular foot that it uses to glide around over the surface. Great. So earlier we saw a slug. Basically the slug would be a snail that has lost its shell, or some of them have actually internalized the shell. Good eye. Tina Molitsova is suggesting that the Anthomastus that we were just looking at there, and we use that kind of as a general term to refer to all those sort of mushroom corals, may actually be the genus Pseudoanthomastus. And so there are particular characteristics of the column and the polyps that she would use to identify those, and she's an expert in that group and has described several species of Anthomastus. We're getting some suggestions on shore that perhaps a collection of a Metallogorgia would be in order. We kind of have defaulted to an assumption that when we see Metallogorgia, we're always seeing the same species everywhere, because where we have collected, the genetics tell us that it is always the same, and it's always the same ophiuroid, but that's never been tested down here in the Central Pacific. So that's something that we'll have on our possible collection list.
Sometimes we're surprised, huh?
Yep. Another one of these, or a couple of these large crinoids, fairly long arms. It's interesting. I see that very often they have their arms just sort of pointed straight up. The yellow one just above it is splayed a little bit more, maybe into a feeding position. Chuck Messing was noting a couple of dives ago for some of the stocked ones, when they had their arms sort of laid out and the whole thing looked like an umbrella, that was a behavior when the currents were really low and the particles were maybe raining straight down. looks like on this rock we have maybe 2 bubblegum corals, one of which is either overgrown with zoanthids, or that could be another kind of sponge, and then behind it is definitely a sponge. So let's check out the 2 on the high part of the rock, please. Understood? Yep. Yeah, so that's exactly what's going on. What you're seeing, that sort of yellow thing in front of the sponge, I believe is a bubblegum coral, much like the red one that we see in the foreground. This is a white morph that has been largely overgrown by zoanthids, which are a colonial anemone. That's the yellow stuff that you see. It's got a very rough texture to the polyp body.
So the white skeleton we're seeing would be from the bubblegum that's
Correct.
sticking out there? Yeah.
Right, so the main axis that's attached to the rock, and then just the tips of the white, that's the actual Octa Coral, and it's being completely overgrown by this zoanthid underneath. I'm hesitating just a little bit because the white in some cases almost looks a little bit like a different kind of zoanthid, but I don't think it is. I think it's a white morph bubblegum coral, and that's not unusual. We see pink, red, and white morphs.
So that's still healthy.
Yep.
Yeah, I see.
Yep.
Eventually, the zoanthid will start to make its own skeleton, or?
I don't think so. Not on the ones that inhabit the bubblegum corals, at least not to our knowledge. It looks like they would prefer either bamboo coral or maybe black coral. So I don't think it's the same species. So they, you have to wonder. I don't know that I've ever seen something that looks like a completely covered, zoanthid that has bubblegum coral underneath it. We always see just a little bit of the tips of the paragorgia still surviving. So I don't know what happens there at the end. It might be in the best interest of the zoanthid not to completely overgrow it, because that's what's
keep it alive. Yeah,
keeping it alive and keeping it held to the substrate. Chris, Kelly, do you want to get a closer look at that sponge? We were at the end of the tether, so we couldn't get any closer in the position we were in.
So Chris just stepped out to grab a hard drive, but I'm sure he would appreciate a close-up, maybe?
I think you're right, Astrid, and we will get him one. And this is a sponge morphology that I recognize. I can't tell you the name right now because I'm not that well versed in the sponges, but I do recognize it, and so I'm going to do just a quick search here. One thing I can tell you while Chris is not here, we collected a sponge earlier, and one of the things that he was really interested in was how it attached to the rock, and he described what was called a lophophytus attachment or a basiphytus attachment. And I noticed as we were zooming in that this is a good example of a lophophytus attachment. What you'll see are basically lots of separate fibers that are attaching it to the rock instead of one clear disc that's more glued to the rock. This is a glass sponge, I can tell you that. You see it's pretty massive, very different kind of morphology than the one that we were looking at earlier that had the vase, an open appearance to it. I'm doing some searching in the background. Unfortunately, have not come up with a name yet. The structure of the database that we're using is such that you have to separately load each family. And if you don't know the family it's in, you have to keep searching. That looks a lot like the ones that we were imaging on the previous dive that had so many different parasites on them.
Oh, that's right,
worms. Is that right? Okay. Well, that'll be interesting to know if we keep seeing that.
we have not have a rock, and I was just about to say you read my mind. If we could see if one of these is loose. Earlier on, I was sort of waiting to see if we saw something else, but I think this is the geology we've got. So if we can grab one of these, that would be great. The bigger, the better, as usual.
Looks like that one might even have a worm tube on it.
Always trying to hone in on the geology samples,
sorry.
I tell you.
See an opportunity, get in there.
I know. You're welcome to pick any life off this rock you want. Yeah, I think any of those up there, that one the lasers are on, the one below, if that's not too big, the bigger would be better. But the one off far to the right that has that looks like an urchin next to it, maybe. That looks a little more angular. I don't. Yeah, you might be right. So just give it a little nudge. Yeah, it looks a little more angular. Yeah, if we can. Okay. Moving on then. It's a 2?
2. And dive 6.
Oh, thank you. Thanks.
In the meantime, I can tell you that I've finally found the right sponge page, and Tina Molitsova has helped me out here, so that last sponge was probably a Polyopogon. Kind of a cool name.
Hi, Scott. I'm back. I had to run up to the office for a few minutes, and I'm trying to spot that sponge on YouTube because Astor told me about it, which would figure. As soon as I step out there, you guys see another glass sponge.
Yeah, Chris, it looks a lot like Polyopogon species A in the animal guide. That kind of massive morphology with a little bit of the shelving around it, if you will.
Okay, I see it, and I agree, Polyopogon.
Yeah, feel free to give it a good squeeze there, just to make sure it's not going to crumble on us. Yeah. Yeah, let's... Okay, yeah, I think that's a pretty good chunk of rock there. Let's go for it.
So this is our first rock sample of the day. We're at 1,833 meters depth. The temperature here is 2.36 degrees Celsius. We're diving today on a feature, on a seamount with the proposed name Keliʻihananui. Keliʻihananui seamount, and that's being named in honor of one of the Hawaiian colonists who passed away on this island in World War II.
Awesome. Thank you much. All right, so we're just going to shuffle pilots around here in a second. So just give us a minute. We'll be on way again. Yeah, our expedition coordinator, Casey Cantwell, just shared a news article from the New Zealand National here with some of our collaborators from New Zealand, Malcolm Clark and Di Tracy. We're working with them as we were coming up through the Cook Islands, so a great news story there. Perhaps we have some listeners out there from New Zealand or the Cook Islands still today. Welcome.
You hit the collect button?
I did hit the collect button.
All right. Seeing we still have sea cucumbers following us, and it's a variety of different crinoids and brittle stars. The sea cucumbers typically are on the sedimented parts, and the crinoids are on the hard parts, although we did see one, I think you may have been at lunch, a spectacular close-up of a sea cucumber that was on the rock that had a very full gut.
Oh, yeah.
You remember that fecal coil that we saw?
Yeah.
Well, we saw who was the culprit.
You saw it coming out? Huh?
No, but we saw it coiled up inside. It was completely transparent.
Oh, wow.
Yeah.
Okay, amazing.
Yep. All right, so we're making our way up this slope again. We're at 1,829 meters. I see a Keliʻihananui-like primnoid Octa Coral. We've seen several of those today. It's right in the lower center of your screen. That's the only coral that I'm seeing in the area. Yeah, we don't need a view of it, pilot. We've seen it several times. I'm just noting it for people on shore. If you do have the time, pilot, there's a sponge on the very lower left of the screen. Thank you. This may be a new sponge for the day. So, Chris, we're going to get a zoom on this. I think it might be a euplectellid, but I'll let you give us your insights.
Yeah, I'm ready. I'm just waiting for a little bit of a zoom.
Yep, it's coming.
I'm uncharacteristically holding back on my IDs.
Yeah, that is uncharacteristic of you. So this is not hollow in the center, Chris. I can see that there's that little point, which is, I presume, the main stalk sticking up in the center of the upper part of the sponge, if that helps you.
Yeah, so what I think we're seeing is we're seeing the back side, or it could be the front side, depending on how it filters, may very well be the front side, of a euplectellid, which you correctly determined, Scott. It's one of the stalked euplectellid glass sponges. It's in the subfamily Bolosomini, and in this one, I think it's actually a Bolosoma. And if we had a view of the other side, you'd see a big scooped out side on the opposite side. This is the round side of it. And then you can see that sort of disc right on the top of it, that circular area. That's the presumably at least where some of the water exits that it's filtering. And you also notice the attachment is directly onto the bottom of the sponge, which is one of the diagnostic characters for these stalked euplectellids. So my best guess is euplectellid in the genus Bolosoma.
Thank you, Chris. Nice explanation. You can see that very defined stalk probably had the basophytus attachment, the hold fast, compared to the previous sponges that we had been seeing, much wider at the base. No, we can keep going. We've seen a lot of these. Thank you. And I'm just going to make a note as we're passing of this yellow sea fan. We did see one of these earlier. This is a possible Paramuriciid, or Paramuricia as a genus, but it's certainly in the family Plexauridae, which is a Gorgonian sea fan. So yeah, I think right now we're interested in getting a little ways up this slope. We're just approaching what was the initial target depth of about 1,800 meters, and we are interested in getting a little higher on this ridge. So unless we see something that is quite different from what we've seen before, I think this is a nice speed.
Agreed.
So when you were out, Del, I was noting, the geology and how we're seeing more of the rock. Are these pillows, do you think, or is this something
I think
else?
a lot of this we've come over, these look like small pillows here on this ridge. And then we can see some rocks that have less sediment over here.
Nice big holothurian again here. This is a sea cucumber, again, a deposit feeder. This may be in the family Synallactidae. Doesn't have a lot of the projections or modified tube feet. Kind of looks smooth, like a flat sole. Munching his way through the sediments. Very colorful. So that one is a lot less transparent than the one we saw. You can't actually clearly see the guts. What we saw was like looking through a window at its entire internal anatomy.
Yeah, we've seen a few of these purple ones already. Oh.
Okay, remember when I said we weren't going to stop?
Well, when you see the
Yes, I am, pilot. Thank you. This is a bamboo coral. And so this is really interesting. So Del, the other day I was saying you could have these whips, and I wondered sometimes that they're forked. Now here's a morphology that clearly you could see could have been a whip for some period of time and is putting out these side branches. So that's why I said I have to stop calling them whips, because it's clear there is at some stage this very sparsely branching thing. Now if we saw this came back in 100 years, maybe this would be a great big bush. And this is part of the problem of trying to identify some of these things from a distance from just the overall colony morphology.
Like six of them there.
Yep.
Yeah.
And so, I would not now try to suggest that these are branches due to some kind of injury. This must surely be just normal branching. It's definitely going to be a bamboo coral, and so one of the things everybody on shore that we want to look for is where are the branchings originating? Does it come out of the white part, the internode, or does it come out of the dark part, the node? And that's in part going to be a clue to what species this is. So what do you think, Del?
I think it's coming out of the dark part.
I agree.
All right.
Yeah, so this would be called a nodal brancher. So that's the first clue to identification, the nodal brancher. So the next clue is, can we see any really obvious sclerites? That'd be the microcalcitic elements. And because we have these mind-blowingly good cameras, we can actually look for that.
That's pretty amazing.
Not normally something you would be looking at 2 kilometers underwater. And I'm not seeing any. I don't see any reflections. Now, I'm sure they're there, but we just can't see them, which suggests that they're really, really small. So this has probably got a lot of scales or very small rods as opposed to needles. The other thing is, remember before we saw these really prominent needles sticking out between the tentacles. Not here at all.
Not here, yeah.
So we've got 3 different characteristics. That's a fantastic, pilot. When the video's happy, that's a lot of information for us. Thank you. So actually, look at this. That's not branching at the node. It's just above it. Great. That's an even better thing to see. I'm also seeing some little white dots at the base of some of these polyps on that left branch.
Right.
Those are eggs. So this is just starting to get into a reproductive status. There's something also feeding on that coral right there. Don't know what that was. Beautiful. Great. So this is not a nodal brancher. This is an internodal brancher, but the branch comes out just above the node. So this could be in the genus Keratoisis. The thing is, with most of the Keratoisis, they should have pretty prominent needles. So this might be another, well, I don't know. I'm hedging my bets here about what clade we're in. I know I can say that it's not C, it's probably not D. Could be another B clade. Beautiful coral, it's really tall. We have a nice Aristeid shrimp swimming by there with its long antennae.
Yeah, National Shrimp Day is coming up, May 10th.
Is that right?
That's right. So.
Wow. That was excellent, pilot. Thank you.
Yeah, you'll see some of these rocks don't have any sediment. These look like some blocks may have tumbled down from up above, whereas a lot of these other ones seem to be more in place, and that's what we'd expect.
Copy that, pilot. Thank you. What we're seeing is a bubblegum coral that's just on the edge of the right of the screen, and another one of these primnoid Caligorgia-like. So we've seen both of these before. So it's just increasing the abundance that we see. We can take a note that these are not so rare that there's only one colony.
I think before today that we'd only seen maybe 1 or 2 bubblegum corals, though, right? Yeah.
Yeah.
We looked for a lot, but
we did. Yeah. And so sometimes we thought we saw them, but they were actually the precious pink coral, the Coralliid coral.
Right.
And it's kind of interesting. We're in the same depth range as we were on. Got to get the names of my seamounts here. Kahalavai seamount, and we did not see them there. So
that's right.
Kind of interesting.
We're not too far away, huh?
And they're certainly not uncommon. Here's another stalked crinoid. I think we imaged one of those before, and there's one of those bamboo corals that's unbranched at this point of its life. Somebody saw a fish. Okay. Oh, there's the fish, just lower left, drifting near the rocks. Right, sorry. Jeez, did it again. Lower right corner. Looks like it's drifting away from us. Yep.
All right. We're still far away, but I'm guessing another Basozetus, another cuskeel.
Looks like the theme of the day, Astrid.
Come on, prove her wrong. Prove her wrong.
Now, Chris, I know you're saying that just because you want to increase the diversity.
That's a Baso.
Yep.
In the ECC. So we joke around a little bit, and of course Astrid is just as competitive as I am, if not more. So it's kind of fun.
Thank you, pilot. We got an idea on that one.
One interesting thing about these Basozetus is that we actually see them with a variety of color morphs. Here we've seen them all very dark colored, but actually a little bit further north, in the very edge of the Clarion-Clipperton zone, we saw actually 4 different color morphs, and we, for now, have put them all in the genus Basozetus. But again, we need to collect these guys to really figure out what they are. But we get some really bright white color morphs and some kind of mottled colors as well. So these guys are still a mystery, but they're very common down here. And I think that's a good kind of indicator with how much we still have to know or have to learn in these environments that, you know, some of the most abundant species that we find out here we haven't even identified.
Well said, Astrid. Yeah, we seem to be rising a little bit more quickly. And we don't have the slope maps available to us right now, but I agree. There's another sea cucumber on the bottom. One thing that is interesting, there's plenty of fairly exposed hard rock, but there aren't that many large sessile corals or sponges for that matter. They've been kind of intermittent.
Yeah.
There's an Anthomastus down here. We looked at one of those earlier, and Tina Molitsova had suggested that the genus was Pseudanthomastus. That's that mushroom coral. Just to the lower, below the lasers on the left side, there's something in the sediment, in a little chute below the rock. I don't know if you're going too fast. It was really small. We can keep going. I'll give you more warning next time. Thank you. I think we're going to see a bubblegum coral on the rock ahead, and it's next to a crinoid, and it gives you a sense of both the scale, the size of the crinoid and the coral. Some of these crinoids can get a pretty good big size. You can imagine if that was perched on that coral. What's on the right, in front of the yellow thing on the lower right of the rock face? It's brown. I'm not sure what's going on there. It might be a sponge. Yep, right directly below the lasers. Exactly. So this might be just a dead sponge, and there's something about the shadow that makes it look like it's on a tripod, but huh.
Yeah.
How interesting. So that is a dead sponge, but look at that, Chris. How's that for an attachment for a sponge?
Yeah, I'm looking at this, and boy, that's unusual.
Yeah.
It's got a whole ton of attachment points there.
Yeah.
And it's almost as though it's a cross between a basiphytus and a lophophytus, but it's definitely basiphytus. You can see the silica plates that are glued onto the rock there, but there's so many of them. It's some type of a euplectellid vase.
It's almost like it's been climbing its way up as it's growing up this rock and, laying down new foundational supports. Thank you, Pilot. That's great. That's really interesting. I've never seen a sponge attached like that.
Is it possible this rock is rolled?
No, this rock is okay. Thank you. So video is wondering if the rock may have rolled or tumbled, and I don't think so. I think it's actually that the larva settled underneath that rock and is growing outwards and then starting to curve upwards. Yeah, oh, there's another one. Yeah, so I was looking over who it was. Annie was just pointing on the right side. There's another one of those same sponges, and it had the same kind of attachments. So if we have the opportunity, this one might be alive. Yeah, right there. No, I think it's also dead, but let's get a quick zoom on that if we could, please. It's now it's above the lasers. Thank you. So we just, this is another theme, Del. We keep saying, we've never seen that before, and then we see it,
we
and
can
then
conjure it up, right?
Yeah, exactly. Yep, look at that. 1, 2, 3, 4, 5, 6, 7, 8. I'm seeing at least 8 different attachment points for this sponge.
Yeah, certainly very different than what we've seen.
Yep.
And otherwise, the structure looks very similar to many of those dead sponges we're seeing a few dives back.
Yep.
Yeah.
It's very open. That's good, Pilot. We don't need a closer view. It was just to verify that it's the same sort of sponge and the same sort of attachment, and we have that now. So it'll be interesting if we find one of these alive just to see what that overall vase looks like. Chris was predicting it was much like the vase that we saw earlier, the Regadrella, that had that sort of sieve plate on the top.
Right, but it looked like it had one attachment. It was on a very flat surface, though, there.
Right, exactly.
There, too.
Yeah,
so.
Here's one right here, actually.
Oh.
So this is the sponge that's just coming in the center there. Gray-white, right at the lasers, yep. So this doesn't have the same kind of attachment points, but it may be another one of the euplectellid vase-like sponges. My sense is it's going to be different from the Regadrella. It looks a much more finer texture. We're way out, but that's my guess right now.
And so if you're just joining us here, it seems like we get close to the bottom, often we'll kick up some of the sediment. Fortunately here it seems to move out of the way with the currents pretty quickly, but that's mostly what you're seeing when the ROV approaches the bottom.
Well, maybe I was wrong. Maybe this is a Rigodrella. All right, Chris.
Well, Scott, I think this actually could be something slightly different here.
Oh, good. That's what I thought at first.
This could be something called a Dictyalis. It's still a euplectellid vase, fairly similar to Rigodrella. But this one has a little bit different pattern in which the osculum, or openings on the sides, are arranged. And as you had already noted, it seemed to have a finer surface texture to it. So I think this is Dictyalis euplectellid vase again.
Thank you. Thank you, pilot. Yeah, we can see the sieve plate looks a little bit different.
Yeah.
Chris, how consistent is the structure of the sieve plate, or does that vary from individual to individual within a species?
I think it can vary somewhat, but you're not going to get in the same species a colony that has it and a colony that doesn't have it. For a particular species, they either have it or they don't have it. And for many years, I was told that the Dictyalis didn't have sieve plates, and then I managed to have some time with Henry Ricewig up in Vancouver, one of the world's authorities on these sponges, and he corrected me on a lot of things.
Thanks, Chris. Nope, we can move on. We've seen that. We've got both a crinoid, the dark red, and a bubblegum coral on the right side, the pink one. Thank you for asking.
So we're getting some questions from Facebook here. We have a moment. So this is one I'd like to know.
Okay, Facebook questions. I'm ready.
Yeah, so one is, how did the brittle star's friend sense his buddy was eating that squid the other day? It did not take him long to get over there.
It did not. So that's a really good question and a good indication of something that a lot of these organisms are doing, and that is that they are using sensory input that is different from visual input. So typically it would be very dark here. We wouldn't have the lights on. The organisms typically couldn't see unless they were seeing another organism that was emitting its own light, so a bioluminescent organism. So probably it was a chemical cue. Another possibility is it's sensing the waves, so it's a kind of like a mechanoreceptor.
Right.
So that ophiuroid clearly made a lot of motion and grabbed something, and so maybe it was setting up
hydrodynamic wave there. Okay.
So it's like, oh, something's going on over there. But more likely, it was chemicals, an odor plume, if you will, being given off by the squid, which was probably the same thing that the first ophiuroid detected when it grabbed it out of the water column, I would imagine.
Yeah, he didn't have to move around too far to find it. He went straight towards it, I thought. So, yeah, great.
And
yeah.
What's good, I don't know if that's somebody picking up on Chris Kelly's nickname for Brutal Star instead of Brittle Star, but...
Right. Yeah, thanks, Jason, for your question.
I'm quite proud right now.
Yeah, it's a good name, Chris. Looks like maybe our Basicetus friend again is swimming along with us. Tim Shank offers that brittle stars can be very good at sensing both pressure waves and chemical sense. So I think my answer was reasonable then. Thanks, Tim. Yep. So it does appear that for whatever reason the C-scribe is not annotating. The C-scribe annotations are not updating on the ship what you're putting in on the shore, so we can't see that. But glad to hear that you guys on shore are putting your observations in. Thank you. Please continue to do so. Nope. Yeah, that's good. Let's just keep scrambling up the slope, and we're happy to do that. Yep. One of the things we're just passing over there, this sort of gray tube, that's a really long stalk from a sponge, which has been long dead. Given these conditions, you know, dead things might persist on the ocean bottom for, you know, decades before they fall apart and disintegrate.
So Les was suggesting yesterday some of the coral rubble we were seeing could have been left over from a wholly different ocean system during the last glacial cycle.
Right,
so,
exactly. We're getting a close-up look at a what we've been calling a Parapodia, bubblegum coral. And I see no reason to change that identification right now.
I don't know. I think it looks a little corallid-like, Scott.
Did you hear me? Did you hear me hesitate, Chris? As we were going in.
I did. I was ready to jump on you.
Yeah, good for you. I think you were right to jump on me. So one of the ways there's a lot of similarities in some respects between the precious pink corals and the bubblegum corals, but the bubblegum corals, where the polyps arise, are often swollen. The branches look like these sort of great big warts or wads of bubblegum, which is how it gets the name, and then the polyps arise from there. This one, you can see the branches were much more smooth, and the polyps were arising directly. So that's probably a coralliid in the family Coralliidae. And there are a couple of different genera, Corallium, Hemicorallium, more than that, but those are the two that we most typically see. And then Roland, our video engineer, pointed out to me that just next to it was another one of the Plesiodiadema urchins, the ones that have the long curved spines, and we saw a lot more of those down below earlier today. I haven't seen quite so many.
Yeah, Scott, this is Tim.
Hi, Tim.
Yeah, did you happen to see any dark masses on that last coral?
Dark masses, no. So I saw the ophiuroid, and maybe I was focused on the polyps. I didn't know what you were referring to.
Yeah, I saw this. I couldn't make out any real form, but it looked like a couple of dark masses up in the upper portions of the branches. Maybe something to look out for the next one.
Okay. Yeah, sorry, Tim. I didn't notice anything that caught my eye that was especially different. It may have been the angle that we were looking at. We're kind of looking on it from above. Yeah, what I'm seeing, Roland is showing me the replay, and I'm just showing areas where the astroskematid arms were wrapped. So I bet that's what you were thinking of as the dark masses. It was just the tips of the arms. There's a metallic gorgia.
Yeah, I don't know.
Well, that's what I see in the replay. So here we're looking at the base of one of those sponges. That's really cool. Now I know what that is. I wouldn't have known if I'd seen it before. I would have thought it was a whole lot of corals or something that had grown together. And then on the top left side of the rock is one of the Metallogorgia colonies. And a crinoid coming into view. We're okay on that brown thing on the rock. We can move to the left. Looks like that mysid that was swimming up there was carrying eggs. There we go. You see that towering Metallogorgia. No, we've seen that one before. Thank you. Several times. And there's some crinoids for some scale. You can see the little tiny stalk crinoids. And one of the possibilities there is there are small stalk crinoids, but also the feather stars, like that yellow one on the front of the rock, begin life stalked. They grow for a while on the stalk, and then they'll break free of the stalk. Yeah. But, you know, I don't know how large they get. They're typically pretty small. So I don't know if those are two stalked crinoids or juvenile unstalked crinoids.
So during the early part of their life cycle, they need to be sessile for some reason? Or?
Correct, yeah. I don't know why that is, but that is exactly the case. There may be a sea cucumber on the top of a pillow lava. Let's see. So it's going to be about 2 o'clock from the lasers. And your lasers are just about to touch the rock that it is on. There you go. And right above the lasers now, there's something purple. Okay. So most of the sea cucumbers we've been seeing have been sitting on sediments. We saw one that was really transparent that was sitting on a rock. Let's see if this is the same as that one. I'm thinking this one might have one of the sails on it. Yep, there we go. So this is actually one of the sea cucumbers. This is referred to as a sea pig, is the common name for this thing. And we often see this one swimming in the water column. You can see at one end there, it's got the sort of a flap or a sail, and it can use that to help it propel itself in the water column just for a short period of time. So this is not the pelagic sea cucumber that we keep talking about. This is a benthic form, but it can change its position, get from one place to another much more quickly by spending a little bit of time swimming in the water column. The left side of this thing is the anterior end.
Okay.
Just to help you out.
That is a help. I would not have guessed that, I must admit.
You can see, well, maybe when we get a little closer, you'll see there's modified tube feet that are kind of frilly around the end, and they're going up and down.
Okay.
And that's picking up sediments from the rock and moving it into toward the mouth. And I can actually see one here that's curled up, and it must be inserted in the mouth. And there's another one that's lifting up right now. And I wonder if there's one being pulled away. It's probably already ingested its sediments. And then the other modified tube feet are to push it around. And then you can see the golden color on the inside. That's part of the intestine.
So that's a future deposit.
Correct, exactly. That is a future sediment deposit. Very good. Thank you, pilot. Very often, you know, when we move by, those sea cucumbers might get caught a little bit in the ROV prop wash, and so then you get to see them doing their kind of flapping, swimming motion. So, Roland, I think that was just like a dark rock face. This might be another one of the sponges that's got the multiple attachments. I really want to see one of these alive now. again, the sessile biology is kind of sparse right here. Not too much.
Yeah, so one of the things we're trying to figure out is the ages of these seamounts. And we dove just a few days ago at Keliʻihananui. We would have thought maybe this would be a similar age to this seamount, given their position, but we didn't see hardly any sediment on the flanks of that one. This one is much more sediment covered. Could just be where we are with respect to the currents, I guess, but definitely a different view and ends up being a different habitat than what we saw on the previous one.
Del, we did get a question from Twitter from a Karen Yip who wants to know how long ago Keliʻihananui may have been at sea level. So was this flat topped, this feature?
Yeah, this feature is a little flat topped. It's not quite that classic kind of guy out shape, but it is a little flat on top. So that was certainly a possibility. We're not going to make it all the way up to the crest, but it's certainly a possibility that it was at sea level and was eroded based on its appearance. And how long ago is sort of a tricky question. We think probably the main pulse of volcanism in here. A lot of things over in the Line Islands region, at least in this part, date back to about 70 million years. But there are a few things that have dated more like 30 or 40 million years. So it'll be interesting to figure this out. One of the interesting things, though, is of course as the seafloor ages, it actually subsides, and it subsides more quickly when it's young. And in this area, the seafloor was probably already about 30, 40 million years old when these things formed. So it would have subsided probably a kilometer and a half since these things first erupted if they were formed 70 million years ago. So exactly how long it was up there depends on how high it was to start with. But maybe not all that long. Because the summit now is, around 1,100 meters or something like that? I'll have to look back. I think so.
Looked like a more concentrated sediment chute as we were lifting off the bottom there. And like here again, if you want to look in camera 2 feed, you can see the camera from Sirius, and you get a better sense of the slope and some of the channels being formed by the way the sediments are being funneled down along the slope.
Yeah, it's one of the interesting things about the Earth. Things tend to self-organize like this, and we see this at lots of scales. We'll see these small channels develop here on the slope of the volcano. When we move up into Palmyra, we'll see these big channels, features out in the middle of the basin. So he's kind of had this idea of things being fractal or looking similar at all scales. Very common for lots of Earth processes and geomorphology and geophysics, and we see this kind of behavior in a broad sense quite a bit.
For a sense of scale, since we don't often see this particular view from the D2 camera, I can tell you we're approximately 3 to 4 meters above bottom as we fly now, so between 12 and 15 feet off the bottom. And we're just making a move up the slope. We're at 1,766 meters depth presently.
Yeah, the ship sort of does a leapfrog with the ROV. Is sort of an interesting dance out here to keep everything in sync, and not trivial when you've got a couple thousand meters of cable running down to an ROV.
One of the things, we've had this discussion a lot among, well, all the scientists. I was going to say the biologists, but all the scientists when we're doing our pre-dive calls and our dive planning calls is how we are moving on the feature, let's say a seamount in this case, a particular ridge of a seamount, with respect to what we believe the dominant currents are in the area. And we talk about being on the lee side or the current side. And for various operational reasons, we're working on the lee side today, and that will be more common throughout the rest of this expedition, which means that the strongest of the currents are going to be on the other side of this ridge that we're climbing. And so I've noted that we see a lot of exposed hard bottom here, but not a lot of suspension feeders. So one possible explanation for that is that although we can see there's obviously current flow, and there's particulates moving through the water column, maybe it's not quite strong enough to have a really high density of suspension feeders. It seems like there's plenty of food. Well, we saw on the bottom, there looked like there were lots of organics on the bottom. So it's not necessarily a question of how much organics, but the rate of delivery of those organics to something that can't actually move around. So Roland's noting that on the right side of the screen here, we see this sort of, I don't know if you call it ropey, but this lumpy sort of material over here, and he's wondering, is that indicative of a lava flow at some time in the past?
Yeah, it very well could be. I mean, some of the finer, smaller scale texture is this crust material, but the gross kind of morphology, we see these, some are pillows, some are more like we call low bite flows. Those are quite common, what we'd expect on the coming down the flanks of a volcano. And so I think, yeah, the overall morphology is definitely volcanic. We're definitely looking at intact rock here, and the sediment is in some places bearing it more than others.
I'm just making some notes in the chat room here so somebody else can make an observation.
Yeah, we were on Keliʻihananui the other day. I even saw some look like lava tubes that had been collapsed. We haven't seen any of that really today yet.
Go ahead, pilot. Copy that, pilot. Go ahead, Nev. I'm listening, Nev. Copy.
So that little white stalk thing to the left of the yellow crinoid is probably one of those juvenile feather stars.
So stand by there on shore. We're just dealing with a technical difficulty here for a minute, so we're going to get up in the water column until that issue is resolved.